Let us discuss the molecular techniques. First, we have the southern blotting and the nucleic acid hybridization. A labeled DNA probe will bind to the specimen if it contains the specific sequence that is being sought. The captured probe is detected by the activity of its attached label. This technique is specific and rapid, but less sensitive than the other methods that involve the amplification steps. Secondly, we have the nucleic acid amplification tests. Nucleic acid amplification tests make the diagnosis by amplifying the specific regions of the genome from the pathogen. Although different methods are used to amplify the pathogen-specific DNA or RNA, the aim is the same: to produce sufficient copies for detection. For example, nucleic acid from the pathogen is separated into single strands, and primers are designed to bind to these target sequences. A polymerase then catalyzes synthesis of nucleic acids, and this process is repeated for multiple cycles. This process is also known as PCR, that is the polymerase chain reaction. Automated systems and commercial kits have made these tests available in many laboratories. Real-time PCR machines measure rising concentrations of target DNA and determine positivity when the concentration passes a set threshold. Nucleic acid amplification tests have the advantage that they can detect slow-growing organisms or those that are difficult to grow, for example, Mycobacterium tuberculosis, or make a diagnosis when samples are rendered falsely negative by the antibiotic therapy. Methods to detect antibiotic resistance genes can also be used to provide surrogate susceptibility results. For example, detection of the RPO beta gene's mutation for rifampicin resistance in Mycobacterium tuberculosis. So these are the basic steps that you're going to follow while investigating a case of infection. I hope you found this piece of information helpful, and for a detailed lecture, stay tuned to sqadia.com.